A warm welcome to everybody who's joined us here in person today. Thank you for attending. And to extend a welcome to the speakers in our next session, which is titled Modeling Rare Disease with Patient-Derived Cells, a Platform for Discovery and Precision Therapies. We are pleased to have a panel of 4 speakers from leading foundations and biotech firms who will share their approaches and insights into this field. Our speakers will discuss how patient-derived cells can enhance the understanding of rare diseases and lead to more precision and effective therapies. So as we come out of the first session where we see the impact of these therapies, understanding how we can keep advancing research, accelerating the path to the therapy to get these important therapies to where they matter most. Just before we start, as a reminder, we'll take questions at the end. We have a Q&A session with the panel. For those online, if you'd just like to enter your questions in the chat, we will get to them during that session. So without further ado, I'll move to our first presenter, who is Dr. Irene Morganstern. Irene is the Director of Preclinical Initiatives at the Children's Tumor Foundation, leading drug discovery in neurofibromatosis, or NF, as I'll refer to it from now on. Irene earned a doctorate in pharmaceutical sciences from the University of Sciences in Philadelphia and completed her postdoctoral training at the Rockefeller University in New York. Before CTF, she worked at generics, designing preclinical drugs for the central nervous system. She has managed data strategy at Regeneron Pharmaceuticals and as part of research and development IT team. Irene's expertise includes preclinical sciences, data innovation, and collaborations to advance treatments for rare disease. Presentation is entitled Accelerating Drug Discovery in NF. Thank you, Irene.
Thank you very much, and thank you for having me here. What a wonderful event. First time attending and speaking, and I'm excited to learn from all the presenters today. So I'll spend the next 20 minutes or so speaking about our work at the Children's Tumor Foundation and how we put all of our effort into accelerating drug discovery for, it's actually neurofibromatosis and schwannomatosis. There are a few reclassifications recently, so hopefully I can clear those up in the next slide or two. For those that have not heard about NF, or collectively, this is neurofibromatosis and schwannomatosis. It's a family of progressive debilitating genetic disorders. 2 main types. There's neurofibromatosis type 1, and then there's a set of schwannomatosis. Within schwannomatosis, there's NF2-related schwannomatosis and non-NF2-related schwannomatosis. Collectively, tumors grow throughout nerves on the body, throughout the body, but that's just a very general way of speaking about it. For NF1 specifically, some of the manifestations could be disfigurement, blindness. Unfortunately, these benign tumors can turn into a malignant type of tumor called MPNST, malignant peripheral sheath tumor. You can have cognitive deficits and bone deformalities. And although it's a rare disease, it's actually not that rare. You can see by the numbers 1 in 2,500 individuals. This is an unmet need. And I'm excited to say, as of last week, we actually have 2 approved treatments now. We had one in 2020 and another one last week. However, for schwannomatosis, there are no approved treatments. Currently, the incidence is lower. And one of the key manifestation in NF2-related schwannomatosis is hearing loss. So the tumor will grow around the 8th cranial nerve, leading to hearing loss and loss of balance, difficulty walking. And again, these are all progressive. Non-NF2 schwannomatosis, these patients have excruciating progressive pain. And again, there's no available treatments. Before I jump into, a few other slides, I wanted to show a video because it's always better to hear it from the patients and learn a little bit about their journey.
My name is Leanna, and I want you to see me and see an F.
My name is Blake, and I want you to see me and see an F.
My name is Jayshawn. I want you to see me and see an F.
People who have NF are so varied in different types.
I basically tell them it's genetic disorder, can cause tumors to grow anywhere on or in the body, and it's like something that you're born with.
I look healthy on the outside, but on the inside I have over 100 tumors in my body.
But what people don't see with my diagnosis is that I'm losing my hearing.
They just see this person, oh, you're really in pain every day?
I have NF1. I'm the only person in my family that has NF.
They make assumptions that they're not just like every other teenager.
Because some people are different than other people.
I might see you as someone who's different, but actually understands you and knows a part of understanding of what you're going through.
All of my tumors are internal. I don't have any skin tumors.
One of the things people can't see about NF is the constant worry we have, knowing that a tumor could be growing anywhere in their body at any time.
I'm not too sure. I might develop tumors, I might not.
I think about the future and I get scared, but then I meet people within CTF and the NF community, and it's inspiring.
Just because I have NF2 and I've had 27 surgeries, despite these challenges, I'm still pursuing my dreams.
Someone could show a scientist, and then they could figure out a cure and keep on experimenting.
Although I have NF and I went through my own struggles, the second I found out my son had it, my entire world changed. And it was my mission from then forward to find a cure or an effective treatment.
The more attention there is to it, the more research, the greater chance they're going to find a cure.
We want everyone to support her and support everyone dealing with NF in the way we've felt so supported by the CTF community and the NF community.
My name is Jesse. I want you to see me and see NF.
My name is CJ. I want you to see me and see NF.
So what we do at the foundation is focus really heavily on drug discovery. So we have been talking about being the drug discovery engine for NF. And so what that means is we'll bring all the right people into kind of one space or, in collaboration. So we have access to patients, to clinicians, to researchers, to government officials. And how can we do this together? How can we bring those folks together to accelerate this process of finding new treatments for NF? And our patients are at the core of everything that we do. And our mission is simple yet complicated, and it is simply to end NF. And so that's a little bit about us. We are in the States, and we also have a headquarter in Europe. So we're in Europe and the US. So we've heard stories already and this morning and all throughout the session. I'm sure we'll hear more about how the patients endure more than a frightening disease, not knowing what will happen, when it will happen, how to treat it, how to handle, giving care to the children, the patients. But the system is also a little bit broken. And we won't get into every part of that system and what's broken and how for rare disease, it's hard to recruit patients. You have multi-sites where you have to get patients from. You have complex protocols, but I will focus on preclinical. That's where I'm heading here. So what are some of the challenges with preclinical drug development? We know it's expensive. We know it is risky. We know that it often does not translate well from preclinical findings into the clinic. We know that some models, and I think I heard it mentioned, they're difficult to get your hands on. So even if you have a repurposed compound and you have a theory, what do you do with that? You need a model. How do you find those models? Sometimes it takes a while to negotiate those terms with institutions to use those models. That leads to very long timelines to initiate a study. Time is of the essence. So whatever we can do to shorten this and make it more cost efficient or effective, that's what we're trying to do here. And of course, standardization. If this is something we're going to use over and over again, these assays, we need to make sure they're standardized and they're used in a similar way. So in order to overcome these hurdles, something that we're working, and this is our newer initiative, it's called the NF-focused preclinical hub. And it's not a physical hub, it's a virtual collaboration kind of concept. And so it's a integrated network of experts, so various institutions, contract research organizations, and other tools. And the idea is to bring them all together into one central virtual space and make them accessible. So how do we make the models available, accessible for drug screening? When I say models, I'm talking about in vitro cell models, in vivo mouse models. We're talking about data tools. We even have pig models we've developed that can be used for PK/PD experiments. And drug libraries, right? How do we, look through, scan the literature? Because unlike other ultra rare diseases, there's a ton of literature around NF. So we've generated NF-focused drug libraries, and how do we source those compounds to make them available for drug screening? So this is our initiative. This is what we're working towards. But how do iPSCs, and that's kind of the focus next, how do they play a role in this? When we thought about which models to generate or build as part of the preclinical hub, we focused on clinical translation. iPSCs were one of the models. We have others. We're also working on PDX models we're trying to generate and put into the hub. But right now we're speaking about iPSCs. And I don't think I have to go into much detail, but we know that these cell types are patient specific, so we can kind of recapitulate the mutations that a patient could have. We can go ahead and differentiate them into disease specific cell types to better understand the pathology in vitro. We can scale them, we can reproduce them. We can even use them for either medium or high throughput screening assays to screen some of the repurposed drugs that we've identified. And they have a translational potential. So here's kind of where, I think that's the button, yeah. So here's where we're fitting in here in this in vitro. We have other models that we're working on, as I mentioned here, but we're just gonna focus in on this little part. Before I talk about how we went about generating these cells, I have to give a little bit of background. So if we focus in, and this is on neurofibromatosis type 1, NF1, the gene is on chromosome 17. And this is a rather large gene, one of the largest in the human genome. It's 60 exons long. And then I think this number is actually outdated, the 2,800 different pathogenic variants. I think there are more reported at this point. And the protein product is neurofibromin, and it acts as a negative regulator of the RAS-MAP kinase pathway. So this is indeed a RASopathy. To have tumorigenesis in the NF1 patients, it's based on a two-hit theory. The first hit is a germline mutation in the NF1 gene here, followed by a somatic mutation in the second allele of the NF1 gene, and you have loss of heterozygosity. This can occur in different cell lineages, causing the complex spectrum of manifestations that I mentioned on the first slide. So it can happen on Schwann cells, astroglial melanocytes, mesenchymal progenitors, and these are just a few examples here. So you can quickly see how complicated this disease is. So how do we model this? How do we even start? What we did here with our first set of iPSCs is based on a publication that came out of University of Alabama at Brigham, UAB, we selected a germline variant based on genotype-phenotype study. This study identified 6 mutation hotspots, and we ended up focusing on this GRD region because this is important for RAS signaling. And not only did we focus on this GRD region, we also focused on a more severe phenotype. This more severe phenotype led to cognitive impairments, spinal neurofibromas. COMs are like pigmented spots on the skin and skeletal abnormalities. So that's how we went about, where do we start with these cells? And we worked with iXCells and their team here to generate our first set of cell lines that will go under this preclinical hub. We used CRISPR, so we did not start with patient generated. There are a few hurdles there we're working through. We started with CRISPR generated cell lines, and we used CRISPR to introduce a missense het knock-in with this arginine 1276 glycine variant, again, identified from this publication. We then used the het cells to introduce a truncating mutation on the stop codon on the second allele. And I'm sure the iXCells team can tell you all about the wonderful work they did to generate these cell lines for us. So right now, we're early on in the process. So I'm afraid I don't have too much data to share. It's just a concept that I'm sharing here. So right now, we are hopeful that these cells will be available through iXCells soon, as soon as they're characterized and validated. And we have big plans for these cell lines. We would differentiate them into, for example, Schwann cells progenitors. These are the main affected cell types in NF1. We would validate these cell lines and develop a high throughput screening platform using them and generate spheroids as well. We do want to go beyond this one specific set of cells. We have plans to look at other mutation types. There are frameshift mutations, splicing mutation, nonsense deletions, so many that we can generate. This is only the starting point. And we absolutely want to expand into schwannomatosis because for schwannomatosis, although the incidence is rather low, I think that's the area that really needs some of these new models. So we're starting here, we're moving forward to all of these other next steps. And for the drug screening approaches, we would go ahead and validate the initial using clinically relevant drugs. As I mentioned, for NF1, there are 2 right now, selumetinib and mirtemetinib, that are on the market. So we would go ahead and start with those validation studies when we have the cells ready to go and generate, next gen 3D models and functional assays. These are our plans, long-term plans here. So, I'll put a quick pitch in for CTF and the preclinical hub, saying that, what we're looking for are vendors or biotech companies that want to work with us and be part of this preclinical hub, industry partners that are interested in NF or that need some of these resources. Again, we're developing these resources for the community, for the research community, for the pharma companies that need to use some of these models. That is our goal, is to make it easier to screen compounds for NF. Foundations with insights, I've already heard so many here that I jotted down, so always reach out. Happy to chat. And institutions that are eager to co-develop innovative models. I think that's about it. And I just want to thank you. We have a small but mighty team at the foundation. And here are most of them. And it really takes a village. And all of these individuals contribute quite, to these preclinical efforts and to the mission of the foundation overall. So just join us in Ending NF. I'm happy to answer questions later or talk about how we can work together and collaborate.